final review. If you are just walking in, make sure you fill one of these out. I'll put them up on one of the first tables back there. This is making a lot of noise too. Oh, cool. Thank you. Yep, that's a lot of noise. Anyhow, so welcome. We can get started. Have you all been able to access the final practice test that I did with no problem? Okay, cool. So here's what I can do for you. I was thinking of maybe trying to go over like a broad review of basically like complement, your innate immune system, B cells, BCRs, maturation, activation, and T cells with TCRs, maturation, activation, and so on, which that's kind of like the bulk of everything. But I don't know, if I do that, I don't know if I'm going to be able to go through the whole thing, right? I will post the answer key right after this. I'm going to go to the office and do it. But if I do the review, I'll basically answer all the questions here. You just got to pay attention, that's all. Or I can just go over this and then talk about the topics as they come up. It's up to you what you would like to do. What's that? I said go over the test. I heard 1 people out of like 30 something. Test? I will post the key, yeah. I mean, we can like, I can go over the test, and as the topics come out, I'll just go ahead and talk about them. Sounds good? Okay, cool. So before I start, some of you have the final on tomorrow. Yeah, that's right. And then some of you on Friday. Oh, really? What time is it? Oh, cool. So it actually worked out for you. Huh? No, well, I thought the final was going to be like in between my review. Oh, well, I'm glad it worked out for everyone. But what I was going to say is don't worry about it, because actually I'm not going to walk because this thing is going to be like bloom, bloom, and it's going to be annoying. The final, it's not that bad. If you've been keeping up with the material throughout the semester, you'll see that a lot of the things get repeated over and over throughout the chapters, right? And if you end up doing an outline of everything, several topics fall under one topic that you just see over and over again, right? So, you know, make sure you study for the final, because it is 100 questions. You do have, you know, B cells, T cells, innate immune system, the GI tract, a little bit of the viruses, because I think he covered part of 11, or he finished it, right? He finished it, okay. So make sure you study, but don't freak out, because a lot of the questions in the finals are more of, are those kind of questions that encompass topics that you see throughout. So for example, you'll have questions asking you about RAG, because you see RAG back to back in like 4 different chapters. About maybe if you think about transcription factors, he'll ask you about NF-kappa B, for example, at some point, because NF-kappa B, you see it at least like 3 times or something like that. Is there any way some of you can close that door for me back there, just because there's a lot of noise? I don't want to. You can come in too if you're coming to the review. Just there's a lot of noise. I want you all paying attention to me. Eyes over here. So yeah, you know, make sure you study, make sure you prepare yourself, but don't panic, because I had a couple of people telling me I'm panicking, right? And it's really not that bad. All right. Any questions before I start? Not really? Okay, cool. All right. So, if like I'm reading something that's like down here and it's not on the screen, just yell at me, stop me. Don't just stay like this because I'm reading and I'm not going to see you. So just tell me. I don't mind. All right? And then for everyone that is just coming in, make sure you grab a evaluation form, which is on that corner. You can do it now or later, but just do it by the end of the review and just turn it in to me. All right? Thank you. So, can all see well? Okay, cool. So, which of the following is not a characteristic of inflammation? An activation of macrophages, increased vascular permeability, vasodilation, pain, and influx of leukocytes. Which one do you think the correct answer is here? A, right? Because inflammation is what our innate immune system and our adaptive to a certain extent uses to fight infections, right? So what was the model that we studied for inflammation? Does anybody remember that? TLR4, does it ring a bell? Right, so what did TLR4 recognize? TLR4 is a PRR or is it a PAMP? Is it a pathogen associated molecular pattern or a pattern recognition receptor? It's a PRR, right? Because it recognizes LPS on the macrophage. The macrophage, so I'm going to write down here. Okay, inflammation. So you're going to have, the model we study was the macrophage getting activated when it recognizes LPS by its TLR4, right? So you're going to have macrophage. Hold on. Macrophage, you're going to have LPS, and you're going to have a TLR4, right? LPS is going to go there. Correct? And then what was the outcome of this? There was a whole molecular pathway you had to study where you had CD14, MBL, IRAC4, and blah, blah, blah. And then in the end, it caused what? It causes phagocytosis, yes, because you recognize a pathogen that's there, it's bad for you and whatever. But what will the macrophage secrete? Cytokines, right? So in the end, you're going to have secretion of cytokines. And remember, does anybody remember what those cytokines were? There's like 5 of them. IL-1, TNF-alpha, IL-1, IL-6, CXCL8, and IL-12, yeah, thank you. Sorry, I'm a little bit hard of hearing, so yell. But yeah, it's all those 5s, right? Those were called pyrogens. They created all these things. Besides the inactivation of macrophages. I'm sorry? Yes, yeah, that's part of the process of fever, but mostly it increases vascular permeability, which means your blood vessels, they get a little leaky. That way stuff from your bloodstream can get in. What would be something that you would like to get into the infected tissue from the bloodstream? Neutrophils, right? Complement too, because you can get complement, C3 getting into the tissue, and then C3 gets activated. You activate complement. You get neutrophils. You get NK cells if it's a viral infection, and so on. But secretion of cytokines, and I'll just write down here, IL-1, IL-6, TNF-alpha, CXCL8, and IL-12, right? Cool. So just to give you a reminder that this is still a thing that you need to know. All right. All right. So the one that's not characteristic, it's inactivation of macrophages, because macrophages are the ones who actually start the whole thing, right? So then for question 2, it's asking, which of the following lymphocytes has a red-orange stained granule and is specialized in fighting parasitic infections? Now this, everybody has to be singing the answer in unison, because pretty sure you ask this every test almost, right? So it's going to be none of the above. Yes, question.
No, I want to
Oh, okay, cool. Because if this was asking about a leukocyte, then it is eosinophil, right? But because we're talking about lymphocytes, it's none of the above, because remember, you start out with a stem cell. The stem cell divides itself into a leukocyte, progenitor, and a lymphocyte progenitor. Right? You also have the one for red blood cells and platelets and so on. But then what will the leukocyte progenitor give, what will it differentiate into to create what cells?
B cells, T cells.
Leukocyte. What? Everything else, right? Besides B, T, and NKs, everything else. It's going to be your monocytes, which then in turn turns into a macrophage. It's going to be your BEN, basophil, eosinophil, neutrophil, your dendritic cells. And then on this side is going to be B, T, and NK. And then over here is going to be BEN, what's the difference between a monocyte and a macrophage? Does anybody remember that? Yeah, the monocyte is circulating in the blood. The NK cell is in your tissue, right? Dendritic cell. Cool. Oh, sorry. Awesome. So, pyogenic refers to the body's production of heat to fight infection. It's false, right? If you can't remember it, the way I used to remember it is that pyogenic has yo. So I would say to myself, and you know, pyo means pus production. So I would say to myself, yo, that's pus, right? So that's the way it sticked with me for some reason. I don't know why. Complement, you should all know it by now, right? Super easy. Do you want me to do go over it really quick?
Yes.
Yeah, I'll do it really, really quick. So complement, the alternative pathway Oh, by the way, sorry for the bad handwriting. I was never able to improve it, so yeah. So the alternative pathway starts with C3. That's soluble, it's floating around, gets hydrolyzed, it turns into IC3, right? And then factor B binds to it. And then this IC3B gets modified by factor D, which cleaves B. So you're going to end up IC3 big B little b. What's this called? This is what kind of convertase? I'm sorry? It's the soluble C3 convertase of the alternative pathway, right? Because it's IC3 big B little b. Then this will cut more C3 that's floating around that's still soluble, but it's going to cut it into C3b and C3a. And C3b, because it has that thioester, very nucleophile bond, it's going to want to bind to something. So it's going to bind to the surface of the pathogen. So then you're going to have C3 gets cleaved by IC3 big B little b into C3b. And you also get C3a, right? And then this will bind to factor B, and then factor D comes in, which cleaves D, right? It's going to form the C3b, big B, little b. So that's the regular C3 convertase of the alternative pathway, right? And this can cut more C3 and make more C3 convertase of the alternative pathway. Or what's the other option? Before that, the C5, right? So if it cuts a C3 and the C3b portion gets stuck to the complex itself, it's going to form the C5 convertase. So C3b, big B, little b plus plus a C3b will yield your C3b2, right? Which this will, do C5, C6, and so on and so forth, right? And this is the C5 convertase of the alternative pathway. Now the classical, super easy. What's so cool about IgM that has to do with the complement in general?
Isn't it the best at activating it?
Yeah, right. It's one of the best at activating because IgM has high avidity and low affinity. It can bind to something that you just recognize, something new, and it can get complement activated while your B cells start going through somatic hypermutation and isotype switching to improve its BCRs, right? But the classical pathway, really quick, you're going to have the C1q binding to, thank you, to either 1Ig IgM, 2 IgGs, or 1 CRP, right? And then once this happens, you're going to get C2 and C4 cut by which subunit of the C1 is the one that has the enzymatic activity? C1s, right? Because the protein is called C1, but you have 3 subunits, QRS. Q binds to one of these, and then once that happens, Q is activated, and in turn it will activate R. R will activate S, and S will start chopping stuff up, right? So in this case, C2 and C4. So C1S cleaves, and then C2A, that's the one we care about. C4b, this is the same thing, C1s cleaves. And you will get the C4b, 2a, which is your classical C3 convertase. And then it's going to be able to cut C3, and then the C3 will feed into this whole system, right? Now, so etc. Right? Those 3 little dots means etc. And like I said, this will feed into this whole pathway, basically. Am I right? Yes? And then if the C4b2a binds to the C3, sorry. if this happens, then you have your C5 convertase, and you just got the MAC activated. All right? If you can't remember it, do this. Write it down repeatedly, and it'll be good for you, because that's pretty much the only way you get to learn this. By the way, I'll have all these papers up here, so you can come take pictures afterwards. Don't worry about it. Or you can take pictures as you go. But going back to the test, for question 4, it's asking, what's the protein that initiates the cascade in the classical pathway? It's going to be C1s, right? Because the initiation is going to be the cutting C4 and C2. And C1s is the one that is doing all the cutting. C1q is going to be the one who binds to the pathogen, right? There's also a slide at the end of the PowerPoint that's named Chapter 3 Slides for Test 1 or something like that. There's actually a nice chart that tells you which proteins are involved, which one starts the pathway, and what do you get as products if you want to review it. It's the one that's named Chapter 3 Slides for Test 1. Yeah, it's like the last PowerPoint you used for Test 1. So then 5, it's asking about the receptor for C3b. Do you all remember? CR1, right? And then what about CR2? It's receptor for what? Huh? C3d. We studied it for B cells, right? And C3d comes from IC3b, which comes from C3b. Right? You remember that? Cool. Toll-like receptor. It's asking which of the following proteins does the toll-like receptor needs to be able to start the cytokine, to be able to start the cytokine cascade. And it'll be, MD2, CD14, and LBP, right? Make sure you go over all these things as you're studying for the final, all these proteins. I don't know if I never see the final. I only see the final when you're taking the test, so I honestly don't know what's going to be in there. But I don't know if he'll go as specific as, which of the following proteins do you need in, for example, toll-like receptor. But it's a good thing if you know it, right? I would definitely focus on those things that get repeated over and over and over again, right? Like, we'll get to it, but like adhesion molecules and BCRs and TCRs, enzymes for like somatic recombination and isotype switching, and all those things that you kind of see the topic back to back on, that you remember you saw it on every test, you definitely want to know it. So for example, complement, was it on every test? Almost, right? Yeah. Didn't he ask at least one question on each test? Oh, okay. Well, it's almost on every test, so you definitely want to know it for this one, for example. So 7 is asking about identifying the mismatched pair. ICAM1 and LFA1. What are these? It's on T cell. What's the function of it, of LFA1 and ICAM1? Adhesion molecules, right? So these are good. CD14 and LPS? Well, this is actually right, because CD14 is the receptor that holds LPS for the toll-like receptor, right? So this is also right. And then NK cells and INF gamma, interferon gamma. Remember, NK cells secreted interferon gamma for virus response, basically. And then all nucleated cells secreted interferon alpha and beta to tell the other cells, hey, I'm being infected by a virus, right? So this is also true. And then selectin with tight binding. This is going back to your neutrophil extravasation. So rolling adhesion, tight binding, diapedesis, and migration. Which step was selectin involved with? Was it rolling adhesion or tight binding? Rolling adhesion, right? If instead of saying, sorry, this is wrong. It's not true. So the one that's mismatched, because this is asking about the mismatched pair, is going to be D. If it said something like immunoglobulin superfamily, or LFA1, for example, again, and tight binding, then it would be right, okay? Catalase is the enzyme used by neutrophils to produce hydrogen peroxide. No, it breaks down, right? It breaks down hydrogen peroxide into water and oxygen, basically. Oh, there you go. INF alpha and INF beta are produced by which cells? Only NK, and only T cells, all nucleated, A and B, none of the above. It's going to be C, right? We just said it. Interferon gamma is produced by NK cells, but interferon alpha and beta, those are used by NK cells to tell the neighboring cells that, hey, I'm getting infected with a virus. Start making changes in your like physiology and your metabolism, so that if you get infected, it's going to be a little bit harder for the virus to take advantage of you. This is chapter 2, I think. Yeah. So if you don't remember it, just make sure you kind of give it a read as well. So going into antibodies, IGs. So let's review the structure of an antibody really quick before I get into this. So an antibody looks like this. Also, my drawing skills aren't that good, so sorry. That's your antibody. Let's say it's a BCR, so it's on the surface of your B cell, right? The antigen is going to bind here. Right? And then we're going to have how many chains in your BCR? So we're going to get, well, how many types? That's what I meant, basically. 2, right? So heavy and light. And what's the genes that code for heavy chain? VD and J. And what else? Because VD and J only codes for your binding site, right? It's basically if heavy, 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 light, 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 light. Just because I don't have any colors on me, but so this is heavy chain and that's light chain, right? So this is the VD and J is going to code for this part, specifically the binding site. But what about the rest? How many types of antibodies do we have? Gamed, GameD, IgG, A, M, ED. So the other gene is going to code for your isotype, right? So it's going to be called C, and then the Greek letter for whatever the antibody is. So mu, delta, gamma, epsilon, alpha. Hold on, let me rewrite this. C, mu, delta, gamma, epsilon, or alpha. Make sense? Do you all remember that? And then the light chain, what's the genes that code for it? V and J, and what else? Because there's also an isotype for the light chain. There was only 2 of them. Kappa and lambda. Remember? And then kappa or lambda. Then you also have your Ig alpha and beta, but those are just always there. They will never change. Okay? In somatic hypermutation, you'll have RAG1 and 2 and TDT on, working all together so that you can select a V, a D, and a J segment and put them together, a V and a J segment and put them together, right? And then during why did nobody correct me? See, I was testing you. You all failed the test. And then during somatic hypermutation, yes, AID. You'll have AID. So TDT, remember, it works by adding nucleotides in between your V, your D, and J, and that's what's gonna create junctional diversity, because it's just going to add random stuff to the middle of your segments, while AID is just going to be point mutations in any part of your V, D, and J, your V and J. So then in the end, your binding sites look a little different, and they're going to either improve, stay the same, or get worse, basically. But those are basically the 3 enzymes you kind of have to remember and its functions, okay? Any questions about this? Yeah, what's up?
Junctional diversity for TDT.
Yeah, junctional diversity for TDT.
What was it for AID?
AID works with somatic hypermutation, and it just does point mutation, so it just switches a C for a U, and then the U gets replaced with something else that's random, and that's just going to give you the mutation. What's up? This is both for BCRs and TCRs. That's a good question, actually. So let's go over the TCR really quick. So your TCR, so I guess I should write here. Your TCR, right, is going to be pretty straightforward, actually. So you either have an alpha, beta chain, right? Your alpha genes will be what? V, D, and J. I'm sorry, VJ. And then your beta genes will be V, D, and J, right? What's the other proteins that will always need to be there next to the T cell, no matter what? I'm sorry? It's kind of like it's the, what? CD3 complex, right? It's the same thing as the Ig alpha, but for the T cell. And it's actually 6 of them instead of 4, instead of just 2 that the B cell has here, right? Instead of being just alpha and beta, you're going to have so this is, and then you're going to have 2 here. What are the 2 that are transmembrane and go inside the cell? Zeta chains, right? And the What's the importance of a zeta chain? Yes, signal transduction. So basically, once the TCR binds to an MHC plus peptide, the TCR needs to send the signal inside the cell so that it can reach the nucleus, and then you can have changes in your DNA expression, right, for activation or repression or whatever. Without zeta chains, that will not be possible, basically, because those are the ones that will send the signals to the inside. Okay, so this is going to be CD3. Ooh. And then zeta chains. Remember, CD3 is 2 epsilons, 1 gamma, 1 delta, and then the zetas is just zeta chains. What's the enzymes that work during somatic recombination in a T cell? Same as the B cells. RAG1 and 2, TDT. Do they go through somatic hypermutation? No, right? Because you need your TCR to be able to recognize the MHC, so you don't want to change that, right? Because remember, we have a set amount of MHCs in our body, and you want your T cells to be able to recognize, I'm not saying all of them, but a few of them, right? And then just to keep going with a refresher, if we talk about MHCs, we have how many types? 2. We have MHC1 that binds to what kind of T cells? CD8 T cells. And we're going to have MHC2 that will bind to CD4, right? The structure, so always make sure you know the structure, right? Because I'm pretty sure he asked me this when I took the class. He asked something like, for MHC1, he asked something like, this is alpha 1, alpha 2, alpha 3, so. Oh yeah, sorry, thank you. So this is the alpha 1, alpha 2, alpha 3 transmembrane chain, and then that's the beta 2 microglobulin, right? And then the MHC2 is actually easier because it's just this with beta 2, right? I don't know if he asked this question, but I remember him asking something like, in the MHC1 protein, which subunit has the transmembrane domain? So basically it's asking which part of the MHC1 binds to the cell, which has the transmembrane domain is going to be this chain that goes from the protein to the cell membrane of the cell. And to be able to answer that, you have to draw it out, or at least imagine it in your head, right? And make sure that you know this. So I always say to make sure that you know your MHCs. And do these have to go through somatic hypermutation or isotype switching? No, because they have nothing to do with BCRs or TCRs, but with BCRs or TCR production. I mean, these bind to your TCR, right? And they are the ones who will be carrying antigens. I'll get to you in a second. Question, who carries MHC1s? What kind of cells in our body? All nucleated cells carry MHC1s, right? Because MHC1s, what are we going to load on an MHC1? What kind of peptide? What's the origin of it? I heard something, but I didn't understand. What? Yeah, right? So it's going to be intracellular stuff. It's going to be like viruses and things like that you show to CD8 cells. And what do CD8 cells do? They're cytotoxic cells. They're there. They're the 008, the license to kill. They're going to go out, look for your infected cells with, flu or whatever, and when they find it, they're going to, make the cytokines rain and induce apoptosis, right? Now, so let me, intracellular. Antigen. And now, what about MHC2? In which kind of cells are you going to find this? Dendritic, B cells, and macrophages. Awesome. Those three are called antigen presenting cells, or APCs, right? And the MHC2s are going to be loaded with what kind of antigen? Extracellular, right? Because it's going to be like a allergen maybe, like pollen, for example, or a bacteria that your macrophage or your dendritic cell are going to take in, right? Process it, present it on an MHC class 2 molecule, and then they're going to go to the T cell and be like, yo, I found this laying around. What do you think? Do we need to create an immune response against it, or we're cool, right? And then the T cell, if some T cell recognizes it, then you get your immune response going, right? So this is going to be B cells. B cells, macrophage, and dendritic. And then extracellular. Awesome. What's the model that we studied of antigen presentation to T cells by an APC? Which cell was the one that was showing the T cell antigens with an MHC2? It had B7, DC sign. Dendritic cells. FDCs, follicular dendritic cells, are completely different from just dendritic cells. Good try though, but make sure you don't get confused with them. And remember that your dendritic cell, goes from the tissue, picks something up, then goes to the lymph node, puts it on this MHC class 2, and then the TCR on a T cell is going to recognize whatever is here, right? And after that, you'll just get activation of your T cell. And then your T cell, depending on what it is, it's going to differentiate into a Th1, which will activate what? Macrophages. Th2, which will activate B cells. Th17, which will call for neutrophils. T follicular helpers that will do what? B cells, somatic recombination and isotype switching regulation. And then Tregs, which will regulate the previous four. And if needed, shut everything down once you take care of the reaction. So this is what I was talking about, how everything is kind of interrelated, connected. And if you can think about this, if you can think about all these topics this way, it's good, because you'll be able to answer the questions more easily. Cool, so let's keep going with the test. What's up?
Do we have to know about the MHC1 and MHC2 types, HLA-A?
Oh, she's asking about if you'd have to know the different types of MHC, like HLA-A, BC, HLA, DQ, DR, and so on. Everything that's on the PowerPoint is fair game, unfortunately. I don't see the test, so I don't know. I mean, it's just like five or 10 different ones out of like many cytokines and things that you need to know. So at this point, if you need to know just a couple more things, it's not a big deal. If I were you, I would know it just in case, right? But again, I can't say I can't say no, because then if it shows up, then it's my fault, right? And then I honestly don't see the test before, so I wouldn't know. Anyways, going back to So now that I talked about all this, all these questions should be super easy, well, most of them, just because it's stuff that I already said. So I'm kind of going to fly through them just so that we have enough time to, for me to keep talking about other topics. But if you have a question, just let me know. I'll stop and I'll go over stuff because that's what I'm here for, and I want you to stop me and ask me questions, okay? So, one binding site of an antibody is composed of 1, 2, 3, or 6 hypervariable regions. So, going back to this really quick, right? One binding site is this. Each heavy and each light chain will have 3 hypervariable HVs, right? Each. So 3 plus 3 is 6. So 6, right? If it was if it asked about one BCR, how many HVs is it going to have? It's going to be 12. If it's going to ask about a secreted IgM that has 5 antibodies all attached together by a J chain, it's going to be 12 times 6, which is 60?
72.
What?
72. 12 times 5 is 60.
Oh yeah, 12 times 5, so 60, yeah. So yeah, make sure you're aware of that. Did he ask about, in any test, about how many HVs were in the IgM? He did? Oh, okay, cool. So yeah, be aware of that, but correct answer is 6. All right. The third hypervariable region of the light chain is determined by, does anybody remember which part of the V, D, and J, well, in this case, V and J because it's light chain, right, codes for the CDR3? So it was going to be the junction. So for BCR, your CDR1 and 2 will always be coded by your V segments. And then your CDR3 in the light chain is going to be your VJ junctions, and then your heavy chain is the same thing. Your CDR1 and 2 are going to be coded by V segments, and then your CDR3 is going to be coded by the VD and DJ junctions. Does it make sense? Yeah? Okay, cool, awesome. This is asking about so whether an antibody is secreted or membrane bound is determined while or during somatic recombination, somatic hypermutation, RNA splicing, DNA splicing. So, I'm sorry? RNA splicing, right? So this is wrong because that's later. But when you're doing when you're producing your BCR, when you work on your DNA, you work on your VD and J and the type of isotype you want to make, right? But when your B cell is choosing whether to secrete the antibody or just leave it as membrane bound, it's something that's going to be chosen in the RNA. And it's basically, in your RNA strand, you're going to have one little part that codes for membrane bound, the other part is going to code for secreted, and you just cut out whatever you don't want. Stick it back together and make your protein, basically. Okay? Enzymes involved in somatic hypermutations is or are RAG1 and 2, AID, RNA polymerase, or C and D. Do we all agree that it's AID? Yeah? Cool. Just remember, if it would ask about somatic recombination, that would be RAG1 and 2, right? So once a T cell has rearranged a TCR, which of the following membrane proteins will the TCR need to leave the ER to get to its final destination, which is your T cell surface, right? And we said that your TCR, there it is, your TCR will always need your CD3 complex and your zeta chains, always. There's no exception to it, okay? So it's going to need, and the CD3 complex is going to be 2 epsilons, 1 gamma, 1 delta, plus your zeta chains. So the correct one is D. Oh, sorry. Sorry about that. Make sense? Yeah? Cool. The importance of the zeta chain is in the key role stabilizing the TCR. No, that's wrong. These proteins will have a role in transducing a signal from the extracellular space to the intracellular space. This looks about right. Then the role in aiding the TCR when interacting with the MHC. Not really, because if you think about where the zeta chains are, they're going to be on the inside, so they don't really help the TCR in binding with the MHC, right? And without zeta chains, ZAP70 would not phosphorylate, and activation of T cells would be impaired. Yep, that's also right. So remember that when your TCR binds to an MHC from up here, basically, right? Your zeta chains will get phosphorylated, which just means activated. And then you're going to have those other proteins that had the chart that you had to study. It was like ZAP70 activates DLC gamma. DLC gamma splits IP2 into DAG and IP3. And then you had to study like the right side of the chart where it said something about calcium going up and calcineurin, and then in the end you'll get activation. So that's what it's referring to basically. So, and this is also true, right? So B and D are correct, which is option E. MHC1 has 2 transmembrane chains with 2 alpha chains and 2 beta chains. Does that sound about right? No. So if you go back, that's MHC2, right? MHC1 is going to have alpha 1, alpha 2, alpha 3, and the beta 2 microglobulin. 1 transmembrane chain, 3 alpha chains, 1 beta 2 microglobulin. Yeah, this looks about right. 1 alpha 1, 1 alpha 2 that will form the peptide binding site. This sounds also right. And has a beta 2 microglobulin that has also a transmembrane chain. And this is wrong. Right? So then the correct answer is going to be B and C. So this is what I say when try to know the structures of everything, right? Because if you can, when you're taking the test, if he ever asks something like that, you can just draw it quickly on the side of the paper, and then boom, you have the answer. So, oh, this is talking about how many different types of MHC genes you can have. So it's asking the max number of HLA genes that Professor Weigel could receive from his parents in the event that he is homozygous for MHC1 locus and heterozygous for his MHC2 genes is, so you're homozygous for MHC1, so you'll have how many from MHC1? 3. And then you're heterozygous for MHC2, so how many can you get? 6. Right? So 3 plus 6? 9. Yeah. Yes. Pretty sure. If anything, just go back on the PowerPoints and review me and double check me, but I'm pretty sure this is the way it is. She's asking about if MHC2, the binding set is going to be alpha 1 and beta 1. But if you can, go to the PowerPoints and check. But I'm pretty sure that's what it is, yeah. Do you all understand this? What's up?
Isn't HLA only for MHC1? 17.
Oh, yeah.
So shouldn't it be 3?
You're right, man.
Oh, that's it.
No, yeah.
Oh, okay.
It's HLA-A or HLA DQ, DR, DS, and DP, something like that, yeah. Thanks for making me notice it. I'm probably change it for next semester because I don't want it to get confusing. But good point. Thank you, though. So then a mutation in the MHC locus will affect the beta 2 microglobulin gene. This is false. The beta 2 microglobulin is not coded with this, okay? Beta-2 microglobulin is just a protein that comes from somewhere else that you just add it on the MHC2 to make it more stable, basically. But it doesn't affect your beta-2 microglobulin. That's a little bit specific. Sorry, yeah, it's false. And I don't know if you need to study this in depth, but he could ask one or two questions that are very, very specific. So it's nice if you get to remember this, or things like this, right? So then, question 19 is asking about the interaction between TCR and MHC. And it says that when interacting with a TCR, the peptide loaded on the MHC will mainly interact with the blank region of the TCR, while the blank and blank regions of the TCR will interact and recognize the MHC. Do y'all remember which parts recognize the MHC? So which parts recognize the MHC of the binding site of the TCR? 1 and 2, and then 3 recognizes mostly the peptide. So if you had to draw it really quick, think of this as your MHC, right? And then think of this as your TCR. Your TCR is going to have CR2, CR, I kind of ran out of space there. But that's kind of how it is-ish, right? So your CR1 and CR2 will recognize mostly your MHC, while your CR3s will recognize the peptide. Okay? There's actually some neat pictures on the PowerPoint if you go back and review it, but that's kind of how it is. Okay? And then what about CD4? If this was MHC2, right? What does CD4 do? Does anybody remember? What's that? It's to help the TCR, yeah. So it's kind of like an arm on the side of the MHC, and it kind of like grabs it from the side, right? And it helps with stabilizing and all that. Cool. Any questions so far? No? Okay. Let's keep going. So we said the CD3 region the peptide will interact with CD3. The CDR3, while CDR1 and CDR2, right? It's going to be B. Then it's asking about SID. So which of the following statements about SID is going to be true? It says that T cells are functional because with SID, TCR recombination is not affected. T cells are not functional because with SID, TCR recombination is affected. One of the RAG genes is affected, which does not influence TCR recombination. B and C are none of the above. It's B, right? Because what does SID do? It affects RAG, and it's just not going to work. More specifically, it does affect one RAG, but this statement is wrong because it says that it does not influence TCR recombination, which is not true, okay? So little Jimmy was taken to the doctor because of an upper respiratory infection. He does not seem to be able to clear. He has chronic respiratory infections. A differential blood cell count showed that Jimmy had normal lymphocyte count, but a genetic mapping of his HLA genes showed that there were several mutations that affected his TAP proteins. Most likely, Jimmy has no opportunity to load peptides on an MHC class 1 molecule, SID, bare lymphocyte syndrome, or A and B or A and C. Which one do you think is going to be? Oh, sorry, my bad. So what do your TAP proteins do? They load MHC1, right? And which disease was involved with TAP proteins? Bare lymphocyte, right? So it's going to be E. All right. So HLA-A will present antigens to a CD4 T cell or a CD8 T cell? CD8, right? Because it's MHC1. So that's wrong and right. Is one MHC1 isotype? Other isotypes include HLA-B and HLA-C. Right? So then it's B and C. Cool. The stages in your B cell differentiations are going to be what? What does it start with? Early pro, late pro, large. So this is wrong. That's wrong because it starts with mature. Late pro, that's wrong because it starts with late pro. So then this is going to be the correct one, right? You'll see, yeah, okay. Also, when you take tests, do this, like eliminate possible answers. If you can, once you read the question, just answer it without seeing. So like cover up your answers and try to answer the question, because sometimes the answer will confuse you on purpose. So if you can answer it before, or if you can write down whatever you know about the whatever it's asking, you're gonna be more likely to answer that question right. Okay. Does anybody do that while taking the test? Yeah. Okay. Good. If not, give it a try. It actually works. So this is asking about the large pre-B cell. What happens as a large pre-B cell in your B cell maturation? It's the first checkpoint. So there, you're going to be done with your with your heavy chain recombination, and you're going to want to start your light chain recombination, but you can't yet because you need to test if your heavy chain actually works, if it can actually hold on to a light chain normally. And you're going to do that by using what? The surrogate light chain, which is formed by V pre B and lambda 5, and those two just join together and make that surrogate light chain. When that happens, what are you going to get after that? Once your first checkpoint, so once your V pre B and lambda 5 can successfully attach to your heavy chain, what is your B cell going to continue to do? You're going to proliferate, and then what else afterwards? What are you missing to make a BCR? You can't make IgM yet. You're missing something of the BCR. Because you made the heavy chain there, but you haven't started on the light chain, right? Because you're right in the middle of your maturation process. So once you do that, you go through your light chain recombination, and then once you're done with that, you're going to make sure and test it and make sure it doesn't react with any self-proteins. And then after that, you turn into a naive B cell that goes to a secondary lymphoid tissue and does it work. It's work. Does that make sense? So when you get questions like this, make sure that you orient yourself in, like Professor Wiggle likes to say, time and space, guys, time and space. Have you ever heard that from him? Yeah, he means it. So you have to, he asks these kind of questions because he wants to make sure that you understand this. So when you answer this, you have to make sure you know where and when you're talking about. So, a large pre-B cell is distinguished by pre-B cell receptor, which consists of a completely functional heavy and light chain with its Ig alpha and beta side chains. What's wrong about this? There's no light chain in this stage, right? It's going to be heavy and surrogate light chain. So then B says a pre-B cell receptor, which consists of a heavy chain and a surrogate light chain with its Ig alpha and Ig beta. This is right? And then the surrogate light chain, the tests whether the heavy chain will be able to bind to the light chain, which is also true, right? So then the correct answer is going to be E. Make sense? Yeah? A mature B cell will choose whether to produce IgM or IgD through RNA splicing. That's the other thing you can do with RNA splicing. The first thing was secreted versus membrane bound. And then the second thing you can do with RNA splicing and BCR is IgM versus IgD, right? And then once you switch to, for example, an IgG, you won't be able to go back to having IgM or IgD because you already cut that part of the DNA out and you deleted it, right? Because you have your VD and J genes, and then downstream you're going to have IgM gene, IgD gene, and then IgG, IgA, and so on. So once you do isotype switch, once you isotype switch to IgG, for example, you cut whatever's in the middle, so you lose IgM and IgD, for example. And you won't be able to come back to it because you lose it, okay? Yeah, so this is going to be RNA splicing. The pre-BCR is also known as the first checkpoint, which is a positive signal that prevents or allows apoptosis. Prevents apoptosis, right? So, because if your pre-BCR works, your B cells get a thumbs up, basically, and it can just keep on going. If it doesn't work, it can go back and try to do another BCR, another heavy chain. But if it messes up twice, is it going to have any more chances? No, it's going to be gone. You're going to have to put the poor thing down, basically, right? Now with light chains, you do have how many tries total? 4. So you have, think about it. We have 2 isotypes, lambda, kappa and lambda, right? And 2 chromosomes with 2 kappas and 2 lambdas each. So you're going to have a total of 4, right, for your light chain. So a translocation of the MIC gene will give you superpowers. That'd be awesome, but no, unfortunately, it's actually kind of sad because you get Burkitt's lymphoma. It won't give you SID because SID is what? It's RAG genes, and then the Behr lymphocyte syndrome is going to be the TAP genes, right? Yeah. Sorry for the dark humor there. But yeah, no superpowers. So prenatal B1 cells, they're going to have little diversity because are they not going to have A, a D, TDT, or are they going to have more diverse V, D, and J junctions? What about TDT? They don't have TDT yet as a B1 cell. Remember, B1 cells are those that won't need T cell activation, right? And they won't have any TDT on them. So you'll have the VD and J and VJ junctions, they will be less diverse, okay? So if this was less right here, it'd be also right. Plasma cells. Who knows what a plasma cell is? What does a plasma cell do? Releases antibodies, right? Who's seen Rambo? Has anybody ever seen the movie Rambo? You know, like when he is like shooting out the machine gun, like, you know, big guy, muscular guy. That's what a plasma cell is basically like a big, angry B cell that's shooting out, instead of, you know, a machine gun, 5.56 ammo, it's going to be antibodies, basically, right? So, but the question is, where can a plasma cell come from, right? Can it come from an activated B cell straight from activation to plasma cell? Which cytokine is going to be involved in telling the B cell, hey, I need you to turn into a plasma cell? IL-10, right? What about isotype switched B cell? Once it finishes isotype switching, can it turn into a plasma cell? Yeah, right? What about when it goes through, when it's a centrocyte and it goes through somatic hypermutation? Can it do that as well once it finishes that process? Yeah? Cool. And what about memory B cells? They can also get activated and then turn into plasma cells, right? Into mini Rambos. So, oh, another question. So you're a plasma cell, right? And the infection is almost done. Your T cell is going to come tell you, hey, stop what you're doing and turn into a memory cell again. What will be the cytokine involved with that signal? IL-4. Cool. Good job. So, 30 is asking about which of the following is not a signal that originates from a successful rearrangement of a beta chain in the pre-T cell receptor, right? So here we're skipping back to maturation of T cells. Remember with maturation of T cells, you work on which chain first? On B cells, you work on your heavy chain first. And what is going to be the chain that looks like a heavy chain in the T cells? Beta. So you're going to work on the beta first, right? You're going to have to make that beta chain first. So activate RAG1 and 2, TDT, somatic recombination. And then once you make it, you have to test it, just like with B cells. When you test your beta chain, it's going to be with what receptor? It's not going to be surrogate light chain. It's going to be surrogate alpha. It's pre-T alpha, it's called as well, right? And then once that happens, your T cell is going to be like, okay, good. You know, this is working. I can go do my alpha chain. And then once it does the alpha chain, what's going to be the next step? What do you want to do before you keep going? You proliferate, and then what are you going to check? If it binds to what? Yeah, so you test if your alpha chain binds to the beta chain, and then what's the next step? What do T cells need to recognize all the time? Because if not, they won't be able to recognize any antigen. MHC, right? So you test your MHC. When you test your MHC, if you recognize an MHC class 2, for example, you're going to turn into a CD4 cell. If you recognize a CD8 cell, you're going to turn into a, if you recognize an MHC class 1, you turn into a CD8 cell, right? So from double positive, you turn into a single positive lymphocyte. And then after that, you want to make sure you don't recognize your own stuff, right? So that's when you go through negative selection, because of whatever recognizes self-peptides, that's when you want to delete that T cell before it causes you an autoimmune disease, right? So another question that you kind of need to know where in this whole process it's asking. So here it's asking which of the following is not a signal that originates from the successful rearrangement of a beta chain in the pre-TCR. So we are right there in the middle of the process, right before positive selection, right? And it's asking it says that one of the signals is stimulate proliferation, which is right. Stimulate expression of either CD3 or CD8 co-receptors. Is this right or wrong? It's wrong, right? Because after your pre-T cell receptor, your pre-TCR, you're going to turn from a double negative T cell to a double positive. That means that you'll have both CD4 and CD8 on your surface. Remember that, okay? So that's wrong. And then stops any additional beta chain locus rearrangement, that's right. And then signals to become permissive for an alpha chain rearrangement, which is also right. So that's the correct answer for the question. Does it make sense? Does anybody want me to go over it again really quick? Yes? So, I don't have Coke. Coca-Cola. And I have to clarify. So so T cell. So T cell maturation, right? You're going to start working on your beta chain. Your beta chain rearrangement, right? With your RAG1 and 2 and your TDT, right? So this is going to be your V, D, and J genes. Once this is all set, you're going to do your pre-TCR check, right? So you want to make sure your beta chain can actually successfully bind later on to an alpha chain. So this is going to be with your pre-T alpha chain, and if this is successful, if your beta chain works, then you're going to stimulate expression of CD4 and CD8. And you turn into a double positive thymocyte. You're going to, and it's all this basically. So these are like all four things. Wait. So you're going to stimulate proliferation, stopping additional TCR beta chain rearrangement. So you're not going to work on your beta chain anymore because it's already working properly. And then you're going to start your alpha chain rearrangement. You're going to become permissive for your alpha chain rearrangement, right? So I'm just going to write here options A, C, and D of question 30, because I'm not going to write all that. Does it make sense? Plus stimulating expression of CD4 and CD8, so you become a double positive. And then after that, you work on your alpha chain, and then your next step is going to be your positive selection, where your TCR will recognize MHC, and it can recognize MHC1. I'm going to write 2 here. And MHC1, right? Sorry, that was. And if it does recognize MHC1, then it's going to become CD4 negative and CD8 positive, because it's going to be MHC1, right? And then for MHC2 is going to be CD4 positive and CD8 negative. Does it make sense? What's up?
So B would be correct if it said both?
Yes, option B would be correct if it said it expressed both CD4 and CD8. Good question, thank you. And then after this, you'll have your, so number 5 would be your broken alpha chain, positive selection, negative selection, where you test T cells against self-antigens. Cool? If you pass this, thymocyte meets prereqs to become a graduated T cell and can go into circulation and go look for a secondary lymphoid tissue to stay there and get activated, basically. Does it make sense? Yeah? Cool. flip back to this? Yeah, I'll keep it here so that you can take pictures of it. Yeah. So, still, question 31 asking about your TCR rearrangement. So, once the first chain of the beta chain of the TCR is rearranged and the cell has finished proliferating, the RAG genes are turned back on. The rearrangement of the alpha chain locus is induced. There is a bigger chance that the rearranging thymocyte will become a gamma delta cell than an alpha beta T cell. A and B are all the above. So is this right? Will the thymocyte have a higher chance of becoming gamma delta than alpha beta? No, right? It's opposite. It's going to be a higher chance of becoming alpha beta than gamma delta. So this is wrong. But then the other two are right, because once the first chain, so once the beta chain is rearranged and the cell finished proliferating after all of your pre-TCR and all that, your RAG genes are going to be turned back on because you're going to start on your alpha chain rearrangement, right? So these two are correct right here. Oh, sorry. Make sense? Yeah. So it's another question that you need to be aware of. Where is all this happening? So 32 is asking about the pre-TCR composed of fully formed TCR with both alpha and beta? No. Beta chain with pre-TCR? Yes. So, yes. The CD3 and zeta chains? Yes. So it's going to be B and C, right? Yes. Cool. Rearrangement of the alpha chain locus continues throughout the process of positive selection and stop whenever a T cell is positively selected. That's true, right? So when you're going through positive selection, when you're trying to recognize an MHC, if your T cell is not lucky and it can't recognize that MHC right away, it'll just try to change a little bit of the alpha DNA, alpha chain DNA, and see if it gets lucky with those changes and recognize an MHC molecule. So alpha chain rearrangement really stops when you finish with positive selection. The T regulatory cell is a type of CD4 cell. Is that true? Cool. Expresses CD25, which is a transcriptional repressor used by T regulatory cells and is unique to these cells. Or is it FOXP3? It's the same sentence. It's going to be FOXP3, okay? And this is, so this is true. What's that? So FOXP3 is just a transcription factor that's unique to Tregs, basically. And they use it to produce genes that will help the Treg do the things that a Treg does, right? So regulate other T cells, basically. Question about positive selection. So positive selection tests whether the blank and blank regions of the TCR are combined to, so what do we test for an MHC? If the TCR combined to self-antigens or MHC? MHC, right? And from the drawing we did, think about it. Which part binds to the MHC? CR1 and CR2. Boom, there you go. So CR1, CR2, MHC. Make sense? Awesome. When the T cell is screening the peptide MHC complex in an APC, antigen presenting cell, the model we studied was your dendritic cell, right? The T cells, LFA1 binds to ICAM1 or ICAM2 on the APC. ICAM3 on the dendritic cell binds to DC sign on an activated T cell. SP1 receptor will be suppressed if the T cell is able to bind successfully to a peptide MHC complex. So this is going back to your T cell activation by dendritic cells, right? So on your yeah. Oh, you're gonna need to speak up. I can't understand. Yes, S1P. Sorry about that. Typo. Thanks for making me notice. I'll change it for next time. But remember that, your TCR will be binding to your MHC. Your T cell is going to have CD28 that binds to B7 in the beginning. You're going to have adhesion molecules and so on. So it's good to know what kind of molecules are where in this scenario, basically. And the correct answer is going to be A and C, right? So the T cell, the LFA1 will bind to ICAM1 or 2 on the dendritic cell or the APC, right? And those are just adhesion molecules. This is wrong because DC sign is on the dendritic cell and ICAM3 is on the T cell. So they're swapped for option B. And then C is true because the SP1 receptor are receptors that will tell the T cells to go away if they can't find anything in that specific area. So if you do find something, you want to turn those receptors off so that the T cell doesn't go away before it can get fully activated, right? Awesome. Any questions so far? Everybody's so quiet today, but it's okay. So which of the following statements regarding the co-stimulatory protein B7 is or are true? The B7 ligand CTLA4 has 20-fold binding strength less than CD28. Is this true or false? It's false, right? CTLA4 will have a much higher binding strength than CD28. So B7 on the dendritic cell binds to either CTLA4 or CD28. Oh, my bad, sorry. CTLA4 or CD28, right? CD28 is going to be there at the beginning, when the T cell needs to be activated and needs to start dividing and producing IL-2, whereas CTLA4 will be there when you need to stop that activation and proliferation because you have enough clones of that original T cell, right? And to be able to stop the whole process, CTLA4 is going to be way much stronger than CD28. So that's why this is wrong. B7 on the surface of the T cell, right? It's on the dendritic cell. So this is wrong too. Make sense? When CTLA binds to B7, the T cell proliferation slows down. That sounds about right. So that's going to be the correct answer. So if you read something like this during the test and you don't know what's going on, just do a little table, right, on the side. You can just do this. You can do T cell, dendritic, and then you can start writing down. So the TCR will bind to MHC, CD28 will bind to B7, CTLA4 will bind to B7 as well. And then if you remember the adhesion molecules, you can also write them down. But that's a good way to answer a question like this. If you start getting confused by the answers, you just do that and then read back and see which one's the correct one. So then 38. A T cell that recognizes a self-antigen will most likely activate and proliferate, right? No. We have mechanisms to prevent that because that would be a autoimmune disease. Nobody wants that. Be rendered anergic because of the presence of B7 on the APC. Will you have B7 on the dendritic cell if the dendritic cell is showing to the T cell something that it picked up that was, let's say, a protein from an epithelial cell, for example, right, which is your own stuff? Or no? Do y'all remember that? So, let me grab a paper. It's really easy. So your dendritic cell, let me write it here like this. your dendritic cell will pick up, let's say like LPS, right? LPS is bad. Hey guys, back there in the corner, if you can, hey, they're not listening to me. Guys, back there in the corner, if you could like keep it down a little bit just because we're trying to do the review. Thank you. Anyways, if a dendritic cell picks up LPS, which is from a bacteria, your dendritic cell will travel to your lymph node. And while it travels, it's going to express MHC2 plus LPS, right? Because that's the thing it picked up. And right next, oh God. Sorry, I'm tired, guys. MHC2 plus antigen, which is the LPS. And it's also going to express B7 because B7 is the molecule that will make the T cell get activated, basically, right? So these are going to be on the surface of dendritic cell. Now, another scenario, the dendritic cell, instead of picking up LPS, it will pick up, let's say, I don't know, selectin. Oh yeah, sorry, my bad. Thank you for making me notice. So let's say it eats some selectin that was just floating around. Selectin is the adhesion molecule for the rolling adhesion, right? So then it will travel to the lymph node because the dendritic cell doesn't know if it's bad or if it's not bad. It's something touches its receptor, it's just going to take it in and then process it, and if it can fit on an MHC2, it's going to go and show it to the T cell, right? So it's going to present MHC2 plus selectin. But in this case, there's no B7. Okay? That's the difference. If your dendritic cell eats something that's your own, then you're not going to have any B7, 0. Okay? That's your sort of like check for T cells that are able to make it through negative selection, right? But they are still dangerous to us because they can still recognize self-antigens, right? Because it's not a perfect, negative selection is not a perfect process, and some rogue T cells can slip through, right? But there's not going to be any B7. So once a T cell recognizes selectin and it wants to get activated against selectin, there's not going to be any B7. So the T cell is not going to be able to do anything, right? So because there's no B7, T cell is rendered anergic, and then after some period of time, the T cell just dies. It's not able to get activated, it's not able to proliferate, and it's not going to be able to induce an immune response against anything. Does it make sense? Yeah? Did I confuse anyone? Nobody wants me to repeat it? Okay, cool. So then, A is wrong because it's not going to activate or proliferate. B will be rendered anergic because of the presence of B7 on the APC. Is that right? No. There's no B7. There's no presence of B7. There's a lack of B7. So will not be rendered anergic because of the presence of B7 on the PAPC. This is a negative, but it's, anyways, the answer is E. Oh, see, the format is all screwed up. I'm sorry about that. But this is also wrong, right? 38, yeah. Because here it says that it will not be rendered anergic because of the presence of B7, right? So if you had an option here saying will be rendered energetic because there is no B7, that would be the correct answer. Okay? Cool. All leukocytes are lymphocytes. Is that true? That's false, right? What about all lymphocytes are leukocytes? That is true. Okay. IL-4 will induce differentiation of what type of T cell? Th1, 2, 17, follicular helper, or Treg? Th2. Awesome. These are just those specific things that as you go over the material, you just kind of have to study it because, and there's no way around it. It's just straight memorization. There's no like logic behind it. It's just that, unfortunately. Leprosy. Do y'all remember leprosy that he talked about? So there's two types of leprosy, lepromatis or tuberculoid. So if you, one time somebody kidnaps you, ties you up on a chair, and then the little thing from Saw shows up and it says, let's play a game. You get to choose whether you get, lepromatis leprosy or tuberculoid leprosy. Which one would you pick? If you had to choose, to make it simple, if you had to choose between getting lepromatis leprosy or tuberculoid leprosy, two diseases, which one do you choose? You get only one choice. Why would you say tuberculoid? Oh, does anybody know why? Or if it's, so if I were you, I would pick tuberculoid. Because remember, leprosy is a bacteria that invades macrophages, and it makes them unable to use their lysozymes, so they will be able to phagocyte stuff, but they won't be able to destroy whatever they take in, right? And these bacteria, they just kind of get into the macrophage, and they have a nice controlled environment. They don't have to worry about anything, and macrophages get inactivated. So lepromatis leprosy will induce a Th2 response, which will cause activation of B cells. And B cells will produce antibodies, but these antibodies won't do anything because the bacteria is actually inside your macrophages. And antibodies won't be able to get into the macrophage, right? While your tuberculoid leprosy is going to induce MHC1 differentiation and Th1 T cells. And what will Th1 T cells do? What do they activate? Macrophages, right? So with tuberculoid leprosy, you'll still get a chance to be able to activate those macrophages that haven't been infected yet and slowly clear that infection. Of course, with treatments and antibiotics and so on. What's up? Th1, that was my mistake, yes. So tuberculoid, bias for Th1, lepromatis, bias for Th2, okay? So if you ever get kidnapped by the Saw guy and it tells you, I want to play a game and you get to choose one of the two, tell him tuberculoid, please. Hopefully it doesn't happen to any of you. Oh, and then the other thing is the infectivity. The lepromatis is more infectious than the tuberculoid. 40. So then the lepromatis leprosy is going to have a high infectivity, and it causes that causes differentiation and bias for Th2 cells, which is correct, right? This is wrong because it says that has low infectivity, which is wrong, and has a bias for Th1 cells, which is also wrong. And then it will have a higher concentration of IL-4, IL-5, and IL-10, which are just the cytokines produced to induce differentiation of Th2 cells, right? So then the correct answer is going to be E. And again, it's a little bit of a specific questions, but it's those kind of things that it's nice to know for the test, because these are specific questions are the questions that will make a difference from getting like a B to an A, for example, right? Yeah, what's up? Yeah, that's what it says here. Okay. So the signal or signals that a macrophage needs to be activated from a T cell are which ones? It was a primary and a secondary signal. I'll just tell you, right? Another very specific question, but the primary signal is going to be INF gamma, right? Which is going to be produced in the infection in general, in the tissue where the infection is going on. And then the secondary is going to be a CD40 ligand and receptor interaction, which will just cause that activation of macrophages from Th1 cells, okay? And all this, if you go back to the PowerPoints with your, the whole T cell mediated immunity, it's all going to be there. I think it's like chapter 8, I want to say. It's one of the last, it's like the 4th test, basically. When an activated naive B cell starts interacting with a T follicular helper, the T cell will release IL-5 and 6 in the medullary cord. IgM will be the most abundant antibody that is being secreted by the beta lymphoblast. B cells will form a conjugate pair with the T follicular helper, which will induce expression of CD40 ligand, and in turn will activate NF-kappa B in the beta cell. Is this true? Right? So it's all of them. I did this so that you have like the 3 things that happen when B cell and T cell interact. It's these 3 things. And then after this, your T cell is going to tell your B cell divide. And then once you have a bunch of B cells ready to fight against the infection, some B cells will be told turn into a plasma cell. And to be able to do that, here's some IL-10. And then the rest are going to be told go into the follicle, form a germinal center, start somatic hypermutation, isotype switching, and then come back with a better, stronger antibody, basically, okay? Which following statements regarding a recently somatically hypermutated and isotype switched B cell is or are true? We'll have to compete to interact with the T follicular helper, which is true. There's a lot of competition in there because you have a bunch of new B cells that just want to show to the T cells, hey, this is my BCR, it's new, it's better. Give me the survival signal, please, I don't want to die. Will die by apoptosis if the B cell is induced to produce BCL-XL? Is this true? No, right? So when the B cell finishes somatic hypermutation and or isotype switching, it's going to need to go to the T cell and, like I said, show that its new BCR is better. When the T cell reads it and it's like, okay, you're good, you know, this new BCR you did is actually better, it's going to signal the B cell to produce BCL-XL, and that's going to be the survival signal. If the B cell for some reason is not able to get to the T cell, or if the BCR is just not good enough, then your B cell is not going to get that BCL-XL, and unfortunately it's going to start the apoptosis program basically and die. So then it can turn into a plasma cell or a memory cell without signals from the T follicular helper. This is wrong, right? Because it needs direction from the T cell all the time. So then the correct answer is going to be A. When the infection just began and we have an increasing amount of pathogens in our body, T follicular helpers are expected to secrete more blank to induce B cells to differentiate into blank. So what do you want B cells to turn into when you have an infection that's just starting now? Plasma cells, right? You Rambo shooting out antibodies. And what do you do to make plasma cells as a T cell? IL-10. Awesome. So this one, C. Make sense? Which of the following regulatory proteins are used to, thank you. Which of the following regulatory proteins is used to slow down the activation of complement? So this is going, jumping back, like we're at the end of the semester, and this is jumping back to the beginning of the semester. So factor P is going to be what? Accelerator, right? It's going to make your C3 convertase go faster. where DAF and MCP are going to be slowing down. So that's the correct answer. Specifically, DAF and MCP, I said CMP before, DAF and MCP are the ones that will stop complement from forming into your own cells, whereas factor H and I are going to be the two proteins that will stop complement into the pathogen surface whenever you have enough complement formed, basically. Which of the following scenarios can be considered a form of passive immunization? So IgA crossing the placenta to enter the fetus's bloodstream, somatically hypermutated and isotype switched IgG that are being released by a plasma cell in the secondary lymphoid tissue during an infection, Steve Irwin getting antivenom after being bit by a black cobra because he felt like petting the thing. Which one's the correct one? I think I had this on the previous test. So it's C, right? It's not A because IgA doesn't cross the placenta. IgG does. Okay? Naive lymphocytes can circulate through the MALT, lymph nodes, spleen, or any secondary lymphoid tissue because they follow a what gradient? CCL 19 and 21, right? CCL 19 and 21, so this is wrong, that's wrong. So CCL 1921 gradient when entering the tissue through the HEV, but once the lymphocyte has been activated in a MALT lymphoid tissue, it will follow a blank gradient and will express the adhesion molecule blank. So it's going to be CCL 25, and I have the same typo as the other tests, alpha 4 beta 7. Okay? Make sense? That's why B cells and T cells in your MALT are stuck in working in the MALT because they just can't go anywhere else, basically. Which of the following molecules is responsible to transport IgG from blood to endothelial spaces? It's all of the above, right? Oh, sorry. Bramble, FCRB, and FCRN. Oh, I did. Sorry, my bad. So when an individual has a Helminth infection, which is basically a parasite infection, which type of response is more effective? Th1 response secreting INF gamma, or Th2 response secreting IL-3 and IL-9, or both are going to be the same thing? It's B, right? So just to give you a refresher really quick, when you have a parasite infection, if you have a Th2 response, you will still get inflammation, which in general inflammation is bad in your GI tract because it gives you diarrhea. Nobody wants that. But you do need to clear your parasites, so that's what you're going to need to do is basically start fleshing out your GI system. But at the same time, you want to make sure that you activate the right parts of your immune system. So what a Th2 response will do is will increase mucus production, so will make your goblet cells work more, and you're going to get more mucus with more like defensins, mucins, and all that stuff. And you're going to tell your eosinophils to start working to fight against this parasite infection, and you're going to tell your B cells to start producing some IgE because IgE works very well with parasitic infections, right? Really quick before I let you go, I'll post the answer key just now, like in 15 minutes. Make sure you, before you leave, please fill out the form that's over there in the corner. You do have to put your PID on it just to prove that you're a student. And it's just to evaluate the whole study union thing, and me, I guess. I don't know. Then one last thing. Make sure you sleep before the test, okay? Because it's no good, like this is one of those do as I say and not as I do type stuff, because I had a final this morning and I went to sleep at 4 and I woke up at 7, which is really bad, right? So don't do that. Make sure you get a good breakfast, that you relax. Make sure you pace yourself. Well, if you have the final tomorrow, then you already have been studying for a while, I would hope so. But if you have your final on Friday, then make sure you pace yourself. You divide, your time so that you can cover everything in a good amount of time. And as I was saying, try to focus on those topics that as you're skimming through the material again, that you see it keeps repeating over and over. Because there's a bunch of things that you keep seeing throughout the chapters that are good questions to ask, because they're going to be like comprehensive questions on kind of everything, right? I'll stick around for a few minutes here if you want to ask me any specific questions. If not, good luck. If you came to my SI sessions during the semester, thank you. And I'll see you around, I guess. Maybe we'll take a class together. Make sure you fill out the form, please. Hey, questions?
Yes.
Cool.
Correct me if I'm wrong,
What's up?
but for number